my name is Sabrina Templo. This is Bio 225 for week 6. Today is June 15, 2020, and this lab experiment is about the environmental influences of microbial growth. And for this exercise, this is the first one, the salt tolerance test. And in this lab, I will be incubating the S. cerevisiae and the S. epidermidis culture broths, which contain different salt concentrations to determine an approximate osmolality range for each microbe. And I already cleared my work surface area, and I also gathered all my material. I did disinfect my area with 10% bleach, and with a permanent marker, I went ahead and labeled all of my sodium chlorides. The first one you can see, this is 1%, 7%, and 15% of sodium chloride. And this I already inoculated with the S. cerevisiae. So let me go ahead and move that over. And then the second one, I'm going to go ahead and demonstrate how I did that with my S. epidermidis. So first, I'm going to go ahead and grab my number 1 sodium chloride. And also take my culture vial of my S. epidermidis, and I'm going to sterilize both of them. And then I have my pipette that's been sitting in alcohol. I have to make sure that it's dry. Then I'm going to collect 2 drops from my S. epidermidis broth. And I'm going to transfer it to my sodium chloride, just 2 drops. Then I'm going to sterilize the lid again of both. And then I'm going to recap it. Now I'm going to grab my 5% sodium chloride, or I mean 7%, and I'm going to do the same thing and sterilize both my sodium chloride and my culture broth to sterilize them. And then I'm going to dry my pipette. Then I'm going to collect 2 drops again of the culture and transfer 2 drops. And then sterilize the lips of both. And then recap. And then after that, I will grab my 15% of the sodium chloride, and I'm going to sterilize both of them again. I'll grab my pipette, make sure it's dry. And then I'm going to collect 2 drops again of the bacteria. And then drop 2 drops into the sodium chloride. Then I'm going to sterilize the lip of both. And then I'm going to recap. So with my 6 sodium chloride tubes, I will put them in my incubation location for 48 hours. After 48 hours, I should see some growth in my tubes. If there is no growth in 48 hours, then I will do an additional 24 hours. If still there is no growth, then that would be a negative result. How I will check for positive results is that I will check for turbidity in any of the broths. So that would mean it's positive. If it's clear, then it would be negative. And then my active cultures, I'm going to put them back in the refrigerator for future experiments. And that concludes my first experiment of lab of week 6 for salt tolerance testing. Thank you.